Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with the epigenetic map. How your life changes your children's DNA. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. Epigenetics encompasses the study of heritable changes in gene expression that occur without alterations in the underlying DNA sequence. This field illuminates the intricate mechanisms by which environmental factors can influence genetic activity. These modifications often arise from various sources, including diet, stress, and exposure to toxins, illustrating how life experiences can leave lasting marks on genetic expression. In this realm, chemical tags, such as methyl groups, attached to DNA and histones, subtly altering gene accessibility. Such modifications can enhance or inhibit the transcription of specific genes, thereby influencing cellular function, and research in epigenetics reveals that the impact of these modifications can extend beyond an individual, affecting subsequent generations. The ability for epigenetic changes to be passed on raises profound questions regarding the inheritance of traits and the biological implications of lifestyle choices. This complexity establishes a bridge between genetics and the environment, emphasizing that both factors play pivotal roles in shaping an organism. Understanding this interplay is crucial for grasping the broader implications of epigenetics, as it highlights the potential for intervention. By modifying environmental factors, individuals may influence not just their own health, but that of their descendants as well. The early 20th century marked a pivotal moment in the understanding of heredity. The discovery of deoxyribonucleic acid, commonly known as DNA, unveiled the intricate mechanisms governing genetic inheritance. It was in the laboratories of prominent scientists that the double helical structure of DNA was identified, elucidating how genetic information is passed from one generation to the next. This structure, composed of nucleotides, serves as a blueprint for the biological traits of living organisms. The foundational work of Gregor Mendel in the mid-19th century laid the groundwork for genetic theory. His experiments with pea plants revealed the principles of segregation and independent assortment, which govern the inheritance of traits. These findings established a framework for understanding how characteristics are inherited in predictable patterns, leading to the formulation of basic genetic laws. As knowledge progressed, the implications of these discoveries began to unfold. Researchers recognized that while genes play a critical role in shaping biology, external factors also exert significant influence. This growing awareness gave rise to the field of epigenetics, which investigates how environmental conditions can modify gene expression without altering the underlying DNA sequence. The realization that life experiences can leave a lasting imprint on genetic inheritance opened new avenues of inquiry, highlighting the interplay between genetics and the environment in shaping future generations. Gene expression serves as a crucial bridge between the genetic code contained within DNA and the resulting traits, or phenotypes, that manifest in living organisms. Within this intricate process, the transcription of DNA into messenger RNA marks the initial step, allowing for the translation of genetic information into functional proteins. These proteins, in turn, play pivotal roles in determining the characteristics and behaviors of cells, tissues, and environmental factors exert significant influence over gene expression, often modulating how genes are activated or silenced. This interaction can occur through various mechanisms such as methylation, which involves the addition of methyl groups to DNA, or histone modification, where proteins that package DNA are chemically altered. These epigenetic changes do not alter the genetic sequence itself, but can profoundly affect how genes are expressed. The dynamic nature of gene expression underscores the complexity of biological systems, where a single genetic blueprint can yield diverse phenotypes based on environmental influences. This flexibility allows organisms to adapt to changing conditions and underscores the evolutionary significance of gene expression. Within this realm, understanding how epigenetic modifications can be passed to subsequent generations expands the scope of genetic research, revealing potential pathways for addressing hereditary conditions and enhancing human health. This new paradigm shifts the focus from static genetic sequences to the dynamic interplay of environmental factors and gene expression. Epigenetics serves as the critical link between an organism's experiences and its genetic legacy. It underscores how external influences, such as nutrition, stress, and exposure to toxins, can modify gene activity without altering the underlying DNA sequence. In early development, these modifications play a pivotal role, guiding cellular differentiation and organ formation. Specific chemical tags, like methyl groups, attach to DNA or histones, influencing which genes are turned on or off at different stages of growth. This regulation ensures that cells develop into the various types required for proper functioning, such as muscle, nerve, or epithelial cells. The timing and nature of these epigenetic changes significantly impact overall health trajectories. For instance, a maternal diet rich in nutrients can lead to favorable epigenetic modifications, potentially enhancing the child's resilience against chronic diseases. Conversely, adverse conditions during critical periods can predispose offspring to various health issues later in life. As researchers delve deeper into this intricate relationship, they uncover not only the mechanisms of epigenetic modifications, but also the potential for interventions that could mitigate the effects of negative environmental exposures. Understanding this complex interaction is essential for advancing the field of developmental biology and public health. The intricate processes of methylation and acetylation serve as crucial mechanisms of epigenetic regulation. Methylation involves the addition of a methyl group to DNA, typically at the cytosine bases, which can inhibit gene expression. This modification alters the accessibility of the DNA to transcription machinery, effectively silencing specific genes without changing the underlying genetic sequence. In this way, environmental factors such as diet, stress, and exposure to toxins can leave their mark on the genome, influencing not only individual development but also the genetic legacy passed to future generations. Acetylation, on the other hand, involves the attachment of acetyl groups to histone proteins, leading to a more relaxed chromatin structure. This modification increases the accessibility of DNA for transcription, promoting gene expression. The balance between methylation and acetylation is critical. An overabundance of methylation can lead to a silencing of essential genes, while excessive acetylation may result in dysregulation of gene expression. These epigenetic modifications are dynamic, responding to both internal and external stimuli. The cellular machinery responsible for adding or removing these chemical groups is influenced by various factors, including lifestyle choices and environmental conditions. This dynamic landscape plays a pivotal role in shaping phenotypic outcomes and highlights the intricate relationship between genetics and environment across generations. Chromatin structure serves as a crucial determinant of gene accessibility, influencing how actively genes are expressed. This complex of DNA and protein undergoes various modifications, which can alter its configuration. When chromatin is tightly packed, genes are typically inaccessible, leading to silencing. In contrast, when chromatin loosens, it allows transcription machinery to access the underlying DNA, enabling gene expression. These modifications can result from environmental factors such as diet, stress, and exposure to toxins, suggesting that the external environment can directly influence chromatin dynamics. This fluidity creates a responsive system, whereby the state of chromatin can reflect an individual's life experiences. In this context, epigenetic marks play a significant role. Methylation and acetylation patterns, for instance, provide a molecular signature that can be inherited, impacting subsequent generations. The implications of these alterations extend beyond the individual, allowing environmental influences to propagate through familial lines. Research indicates that these changes can impact various biological processes, from development to disease susceptibility. This suggests that understanding chromatin architecture and its regulatory mechanisms is essential for deciphering the complexities of heredity and the interplay between genetic predisposition and environmental factors. The exploration of these dimensions underscores the delicate balance inherent in biological systems, revealing a rich interplay between structure and function. Environmental factors exert profound influences on epigenetic markers, shaping gene expression across generations. This realm encompasses a variety of elements, including diet, stress, and exposure to pollutants. In this context, nutritional intake plays a critical role, where specific nutrients can alter chemical modifications to DNA. The presence of methyl groups, for instance, may be enhanced by folate-rich foods, thereby influencing gene regulation. Stressful experiences also leave their mark on the epigenome. Chronic stress can lead to alterations in the expression of genes associated with stress response, which may be inherited by offspring. This phenomenon reflects a biological adaptation to environmental demands. As organisms strive to balance survival and reproduction in a changing world, pollutants present another layer of complexity. Exposure to harmful chemicals can result in long-lasting changes to the epigenetic landscape. These alterations may predispose future generations to various health conditions, illustrating the intersections of environment and biology. The epigenetic map is not static, but is instead a dynamic representation of one's life experiences. Each modification contributes to a broader understanding of how life circumstances can transcend individual existence, influencing not just the immediate organism, but also its descendants. This intricate web of interactions emphasizes the significance of safeguarding environmental health for future generations. Nutrition serves as a pivotal component in the realm of epigenetic modifications. The substances consumed not only provide essential nutrients, but also influence the molecular mechanisms that regulate gene expression. Specific vitamins and minerals, such as folate, B vitamins, and certain fatty acids, play critical roles in DNA methylation and histone modification processes. These modifications can alter how genes are turned on or off, thereby affecting various physiological traits and health outcomes in offspring. In this context, a diet rich in whole foods, including fruits, vegetables, and lean proteins, can foster a favorable epigenetic environment. Conversely, diets high in processed foods and trans fats may lead to adverse modifications. The implications of these dietary choices ripple through generations, as the epigenetic tags placed on genes can be inherited, shaping the health profiles of future generations. Research indicates that maternal nutrition during pregnancy significantly influences fetal development and long-term health. The consumption of nutrient-dense foods can establish a foundation for optimal growth and metabolic function, while deficiencies or imbalances can predispose descendants to various health challenges. This relationship between nutrition and epigenetics underscores the importance of informed dietary choices, not only for individual well-being but also for the potential impact on lineage. Each meal represents a choice that reverberates through time, influencing the genetic legacy passed down through generations. Stress serves as a potent modifier of gene expression, leaving indelible marks on the epigenetic landscape. Adverse experiences, whether acute or chronic, can trigger biochemical pathways that alter the way genes are regulated. This region of study reveals that emotional and psychological stresses activate responses that might not only affect the individual, but also reach across familial lines. Biochemical changes induced by stress can lead to the addition or removal of chemical tags on DNA, such as methyl groups. These tags act as switches, turning genes on or off, thereby affecting traits, behaviors, and health outcomes in subsequent generations. The ramifications of these alterations can emerge in offspring, who may inherit predispositions to anxiety, metabolic disorders, or other health conditions. In laboratory settings, models have demonstrated that the offspring of stressed individuals often display altered stress responses and behavioral tendencies, emphasizing the lasting influence of parental experiences. These findings underscore the intricate interplay between environment and genetics, showcasing how trauma or stress can weave through generations. Understanding this relationship opens avenues for exploration in both psychology and medicine, guiding interventions that may mitigate the effects of adverse experiences. This perspective encourages a holistic view of health, emphasizing the importance of nurturing environments for future generations. The epigenome responds to various environmental factors, including toxins and chemicals, which can alter gene expression without changing the underlying DNA sequence. This region of biological activity is particularly sensitive to external influences. Prolonged exposure to pollutants, heavy metals, and synthetic compounds can induce chemical modifications. These changes may silence essential genes or activate harmful ones, thereby influencing the health of not only the individual but also successive generations. Substances such as bisphenol A, phthalates, and pesticides have been identified as significant disruptors of epigenetic regulation. These agents can lead to aberrant DNA methylation patterns, which are pivotal in controlling gene activity. The consequences can manifest in various forms of disease, including metabolic disorders and neurodevelopmental issues. Moreover, maternal exposure to such pollutants during critical periods of fetal development raises concerns regarding transgenerational effects. The potential for these modifications to be inherited means that the health of future offspring may be compromised by environmental exposures experienced by their parents. Studies have shown that even brief encounters with certain chemicals can leave lasting marks on the epigenome, underscoring the fragile interplay between biology and environment. This intricate relationship highlights the need for vigilance in monitoring and regulating exposure to harmful substances to safeguard both current and future populations. Transgenerational epigenetic inheritance refers to the phenomenon where changes in gene expression, influenced by environmental factors, are passed down to subsequent generations. This complex process raises intriguing questions about the extent to which experiences, behaviors, and exposures can alter the genetic landscape, not only for individuals, but also for their descendants. Research suggests that modifications such as DNA methylation and histone modification can affect how genes are turned on or off, potentially leading to inherited traits that reflect ancestral experiences. Through inheritance, scientists navigate a landscape filled with both concepts and controversies. The stability of epigenetic changes across generations is an area of ongoing investigation. Some findings indicate that certain modifications may persist, while others appear transient, fading after a few generations. The implications extend into areas such as health, behavior, and even susceptibility to diseases, raising ethical considerations about the manipulation of epigenetic markers. Critics often question the extent of these effects, debating whether they can be reliably observed in humans or if they are primarily evident in model organisms. This skepticism fuels discussions within the scientific community, emphasizing the need for rigorous research to substantiate claims. Understanding transgenerational epigenetic inheritance may reshape perceptions of genetics, shifting focus from static DNA sequences to the dynamic interplay of environment and heredity over generations. The Dutch hunger winter represents a pivotal case study in transgenerational epigenetic inheritance, illustrating how extreme environmental conditions can leave lasting marks on the genome. During this period, from 1944 to 1945, food scarcity profoundly impacted the population of the Netherlands. Individuals exposed to severe malnutrition exhibited alterations in their epigenetic markers, particularly in genes associated with growth and metabolism. Subsequent research revealed that children born to mothers who experienced these conditions displayed not only physical health issues, but also variations in DNA methylation patterns. This form of epigenetic modification, often influenced by nutritional status, had implications for their metabolic health and susceptibility to chronic diseases later in life. They carried within their cells the echoes of their parents' suffering, a biological record of adversity. Further exploration into the offspring of those affected unveiled a multi-generational aspect of these changes. Grandchildren of those who experienced the hunger winter exhibited different health profiles compared to their peers, suggesting the potential for epigenetic modifications to transcend generations. These findings underscore the profound impact that environmental factors can exert on genetic expression, challenging the notion of genetics as a fixed entity. The Dutch hunger winter serves as a stark reminder of how human experiences can resonate through time, intricately weaving into the very fabric of genetic inheritance. Biological mechanisms underpinning epigenetic inheritance reveal the profound ways in which life experiences can alter gene expression across generations. Chemical modifications to DNA, such as the addition of methyl groups or the alteration of histone proteins, play a crucial role in regulating gene activity without changing the underlying genetic code. This process allows environmental factors, ranging from nutrition to stress, to influence how genes are turned on or off. In this intricate system, small RNA molecules also contribute by guiding the cellular machinery in silencing or activating specific genes. Such dynamic regulation ensures that an organism can adapt to its surroundings, promoting survival and resilience. When these modifications occur in gametes, the potential for transgenerational effects emerges. Offspring inherit not just the genetic code but also these epigenetic markers, which may predispose them to certain traits or health conditions. Research continues to uncover the complexities of these interactions, showing that experiences such as trauma or exposure to toxins can leave lasting marks on DNA that may persist in subsequent generations. This region of study highlights the intricate interplay between environment and biology, emphasizing that the implications of one generation's experiences may ripple through the DNA of many to come. Each discovery deepens the understanding of life's continuity, revealing layers of influence that shape existence itself. The interplay between environment and health emerges as a vital component in the study of epigenetics. Changes in lifestyle, diet, and exposure to toxins can alter gene expression without modifying the underlying DNA sequence. This modification creates a cascade of effects, evitos, influencing susceptibility to diseases such as diabetes, cancer, and cardiovascular disorders. It becomes evident that these shifts are not merely individual experiences. They can reverberate through generations, impacting descendants in profound ways. Research illustrates how environmental factors can lead to chemical modifications of DNA, influencing gene activity. For instance, stress and malnutrition can impose lasting marks that affect cellular function. These changes often manifest in health outcomes, creating a lineage of predisposition or resilience. The implications extend beyond the individual to encompass family health histories, emphasizing that inherited traits are not solely determined by genetics. In certain populations, patterns emerge linking specific environmental exposures to increased disease prevalence. This region, once untouched, now bears the burden of industrialization, illustrating the stark reality of human impact. Each revelation emphasizes the importance of understanding epigenetic mechanisms in public health, as interventions may mitigate risks that span generations. As this knowledge expands, it becomes increasingly clear that health is not an isolated journey but rather a continuum shaped by both choices and circumstances. The intricate interplay of epigenetics significantly influences the development of cancer, changes in gene expression, rather than alterations in the underlying DNA sequence, can initiate the processes that lead to malignancy. Environmental factors, lifestyle choices, and even stressors have the capacity to modify the epigenetic landscape. Methylation patterns and histone modifications serve as key players in this transformation, regulating the activity of oncogenes and tumor suppressor genes. The accumulation of epigenetic alterations over time can result in the activation of pathways associated with cell proliferation and survival, while simultaneously silencing mechanisms that would normally induce apoptosis or cellular repair. This disruption fosters an environment conducive to uncontrolled growth. Specific exposures, such as tobacco smoke or certain chemicals, have been documented to induce such changes. In certain instances, these epigenetic modifications can be reversible. Therapeutic strategies targeting the epigenome are being explored as potential interventions to restore normal gene function and inhibit tumor progression. As research continues to advance, the understanding of epigenetic mechanisms in various cancers deepens. By unraveling this complexity, scientists aim to enhance prevention strategies, early detection, and personalized treatment approaches. The passage of time brings profound changes at the molecular level, influencing not only individual health, but also the genetic legacy bequeathed to future generations. Epigenetic modifications accumulate throughout life, altering gene expression without changing the underlying DNA sequence. These alterations manifest in various ways, including changes in cellular function and increased susceptibility to diseases. As years progress, certain genes may become permanently activated or silenced, reshaping the biological landscape. Aging introduces a gradual decline in cellular mechanisms, leading to a phenomenon known as epigenetic drift. This drift often results in altered patterns of gene expression, impacting processes critical to health, such as inflammation and metabolism. The intricate interplay between environmental influences and genetic predispositions further complicates the aging process. Factors such as diet, stress, and exposure to toxins leave marks on the epigenome, potentially affecting offspring. In this context, the understanding of epigenetic effects extends beyond individual experiences. It raises questions about how lifestyle choices and environmental factors can influence not only personal health, but also the well-being of descendants. By examining the epigenetic landscape shaped by aging, researchers seek to uncover the mechanisms that govern these profound changes, offering insights into the enduring impact of time on our biological heritage and the health trajectories of future generations. Unki experiences play a critical role in shaping gene expression, a phenomenon explored extensively within behavioral epigenetics. This branch of science investigates how environmental factors, lifestyle choices, and emotional states modify the epigenome, influencing the way genes are turned on or off. Individuals exposed to varying conditions, such as stress, nutrition, and social interactions, exhibit distinct patterns of gene regulation, which can persist across generations. Research has shown that traumatic experiences can lead to epigenetic changes that affect stress response and behavior. For instance, offspring of individuals who faced adversity may inherit altered gene expression profiles, predisposing them to similar challenges. These modifications are not merely biochemical, they can manifest in behavioral traits, suggesting a biological transmission of experience. Nutritional influences also serve as a key component. Dietary choices can induce epigenetic modifications that affect metabolism and health outcomes. The implications extend beyond a single generation, as these changes may alter the epigenetic landscape of descendants, thereby shaping their responses to environmental stimuli. The intricate relationship between experiences and gene expression underscores the complexity of biological inheritance. Insights from behavioral epigenetics reveal that the legacy of individual lives is intricately woven into the fabric of future generations, highlighting the profound interplay between biology and experience in shaping human health. The microbiome plays a pivotal role in epigenetic regulation, influencing gene expression through complex biochemical interactions. This diverse community of microorganisms residing within the human body interacts with host cells, facilitating metabolic processes essential for health. Variations in the microbiome can trigger epigenetic changes, modifying how genes respond to environmental stimuli and lifestyle choices. Certain microbial metabolites, such as short-chain fatty acids, can impact histone modifications and DNA methylation patterns. These alterations may affect the expression of genes associated with inflammation, metabolism, and even behavior. The dynamic relationship between the microbiome and its host underscores the significance of diet, lifestyle, and microbial diversity in shaping health outcomes. In this context, the environment serves as a critical mediator, determining the composition of the microbiome. Factors such as nutrition, antibiotic use, and exposure to pathogens can induce shifts in microbial populations, subsequently influencing epigenetic mechanisms. The ramifications of these changes extend beyond the individual, potentially influencing offspring through a process known as transgenerational epigenetic inheritance. The intricate interplay between the microbiome and epigenetics exemplifies the complexity of human biology, emphasizing how external influences can leave lasting marks on genetic expression. This emerging field of research continues to unveil the layers of interaction that govern health and disease across generations, highlighting the multifaceted nature of human existence. Maternal influence plays a crucial role during pregnancy, where the environment experienced by the mother can induce epigenetic changes that affect the developing fetus. Various factors, such as nutrition, stress, and exposure to toxins, have been shown to leave lasting marks on DNA, modifying gene expression without altering the genetic code itself. This period becomes a vital window of opportunity, as the fetus is particularly sensitive to maternal conditions. Research indicates that a mother's diet during pregnancy can lead to modifications in the offspring's metabolic pathways. Nutrient availability shapes not only immediate health but can have repercussions extending into adulthood, influencing susceptibility to conditions such as obesity and diabetes. Stress experienced by the mother may also trigger epigenetic alterations, potentially affecting the child's stress response systems. These changes do not merely vanish after birth. They can persist throughout life, establishing a biological framework that can influence future generations. This region of study underscores the importance of maternal health and well-being, as the implications stretch far beyond the individual, reverberating through familial lines. The interconnectedness of life experiences and genetic expression invites a deeper understanding of human biology and the factors that shape it. Epigenetic modifications thus emerge as a bridge between environment and heredity, a silent dialogue unfolding across generations. Paternal contributions play a crucial role in shaping offspring health, with sperm epigenetics serving as a key mechanism. This region of inquiry reveals how environmental factors can influence the epigenetic landscape of sperm. Research indicates that specific epigenetic marks within sperm can alter developmental trajectories. These marks can dictate cellular processes such as growth, metabolic functions, and even predispositions to diseases. Observations show that paternal lifestyle choices, including diet and exposure to toxins, may lead to enduring changes in the epigenome of sperm cells. This highlights the intricate connection between paternal health and the well-being of future generations. Recent studies demonstrate that when these epigenetic alterations are present, they can affect not only the immediate offspring, but also subsequent generations, creating a continuum of inherited traits. The implications are profound, suggesting that the paternal environment creates a legacy that extends beyond mere genetic transmission. In this context, the exploration of sperm epigenetics continues to unveil the complexities of heredity, providing insight into the biological narratives woven into the fabric of life. The intricate mechanisms of epigenetics play a significant role in the development of neurodevelopmental disorders. These conditions, which include autism spectrum disorder and attention deficit hyperactivity disorder, are influenced by a myriad of genetic and environmental factors. Understanding how epigenetic modifications affect gene expression during critical periods of brain development offers valuable insights into these disorders. Research indicates that environmental exposures, such as pollutants and nutritional factors, can lead to alterations in the epigenetic landscape, thereby impacting neural pathways. Methylation patterns, histone modifications, and non-coding RNAs are pivotal in regulating genes essential for neuronal growth and synaptic plasticity. Changes in these epigenetic markers can disrupt normal brain function, leading to the manifestation of behavioral and cognitive challenges. Animal models have demonstrated that alterations in epigenetic regulation can result in neurodevelopmental anomalies, emphasizing the importance of early intervention. The timing of environmental exposures during sensitive developmental windows is crucial, as epigenetic marks established early can influence neurodevelopment long after the exposure ceases. This field of study continues to progress, revealing potential targets for therapeutic intervention. By understanding the epigenetic factors contributing to neurodevelopmental disorders, researchers aspire to pave the way for innovative treatments that address the underlying biological mechanisms, potentially alleviating the burden on affected individuals and families. Investigations into mental health reveal a complex interplay between genetic predispositions and environmental influences. This region of study underscores the significance of epigenetics, which modifies gene expression without altering the underlying DNA sequence. Factors such as stress, nutrition, and childhood experiences can leave lasting marks on an individual's genetic profile, influencing mental health outcomes across generations. In examining these interactions, researchers have identified specific epigenetic modifications associated with conditions such as depression, anxiety, and schizophrenia. These alterations can affect neurotransmitter systems, neural plasticity, and stress response, further complicating the landscape of mental health. The cumulative effects of early life stressors or trauma may trigger epigenetic changes, leading to heightened vulnerability in offspring. This phenomenon illustrates the potential for transgenerational transmission of mental health risks. When one generation faces significant psychological challenges, their descendants may inherit not only genetic factors, but also epigenetic modifications that predispose them to similar conditions. Unraveling these intricate connections holds promise for targeted interventions and preventive measures, aiming to disrupt cycles of mental health disorders. Understanding the dynamic relationship between genes and the environment provides valuable insights into the biological underpinnings of psychological resilience and vulnerability, helping to illuminate pathways toward effective therapeutic strategies. The potential of epigenetic therapeutics lies in their ability to modify gene expression without altering the underlying DNA sequence. This approach opens avenues for treating a range of conditions, from cancer to neurological disorders. By targeting specific epigenetic marks, it is possible to enhance or silence gene activity, offering a refined method of intervention that aligns more closely with individual biological needs. Research in this area has produced promising results, revealing that specific compounds can reverse harmful epigenetic modifications. These modifications may arise from environmental factors such as stress, diet, and exposure to toxins. By restoring healthy gene expression patterns, these therapies aim to alleviate symptoms and improve overall well-being. Challenges remain significant. The complexity of the epigenetic landscape demands a comprehensive understanding of how various modifications interact with one another. Off-target effects, where unintended genes are affected, pose risks that must be meticulously managed. Additionally, the long-term implications of altering epigenetic marks are still being studied, necessitating caution in clinical applications. Regulatory frameworks are evolving to accommodate this emerging field, balancing innovation with safety. The integration of epigenetic information into personalized medicine represents a frontier in healthcare, where tailored therapies could adapt to both the genetic and environmental contexts of individuals. Continued exploration in this domain promises to deepen understanding and enhance therapeutic efficacy. Lifestyle choices significantly influence the epigenome, shaping the expression of genetic material across generations. Diet, for instance, plays a crucial role, with specific nutrients impacting the addition or removal of chemical tags that modify gene activity. This region of study reveals that a nutrient-rich environment fosters optimal epigenetic modifications, while a diet deficient in essential compounds can lead to detrimental changes in gene expression. Physical activity also contributes to these modifications. Regular exercise has been shown to alter the epigenetic landscape, leading to improved metabolic health and a reduced risk of chronic diseases. These changes may not only affect the individual but can extend to offspring, conveying a legacy of health or risk. Stress, conversely, has a profound effect on epigenetic markers. Chronic stress can initiate a cascade of molecular changes that may predispose future generations to anxiety and other mental health disorders. The mechanisms underlying this transmission are complex, involving interactions between genes and environmental factors. Exposure to toxins further complicates this landscape. Pollutants can induce epigenetic alterations that may manifest as health issues in both the exposed individual and their descendants. Such insights emphasize the intricate relationship between environment, lifestyle, and genetic expression, underscoring the potential for preventive measures to shape future health trajectories. An epigenetic reprogramming presents a significant frontier in regenerative medicine, offering pathways to modify cellular behavior without altering the underlying DNA sequence. This approach involves altering the epigenetic marks that govern gene expression, potentially rejuvenating aged or damaged cells. Such processes can restore cellular function, making them more akin to their youthful counterparts. The implications for therapeutic applications are profound, enabling targeted interventions for a range of degenerative diseases. Research indicates that specific environmental factors, such as diet and stress, can induce epigenetic changes that enhance or inhibit cellular repair mechanisms. It suggests that reprogramming can be harnessed to manipulate stem cell differentiation, guiding them towards specific lineages that can repair damaged tissues. This method may hold the key to treating conditions like heart disease, neurodegenerative disorders, and various injuries. The ability to influence epigenetic states raises ethical considerations, particularly regarding long-term consequences and the potential for unintended effects. Monitoring these changes within a clinical context becomes essential, ensuring that interventions promote health without adverse repercussions. As this field progresses, understanding the delicate balance between intervention and natural epigenetic regulation will be crucial. Advancements in techniques for reprogramming could lead to breakthroughs that redefine regenerative capabilities, fostering resilience within the human body. Exercise functions as a powerful modulator of epigenetic factors, influencing gene expression in ways that extend beyond immediate physiological changes. It initiates a cascade of biochemical responses that can alter DNA methylation patterns and histone modifications, shaping the epigenetic landscape. Regular physical activity has been shown to increase the expression of genes associated with muscle growth, endurance, and overall metabolic health. In the context of epigenetics, these alterations are not limited to the individual. They can be transmitted to future generations, suggesting that the benefits of exercise may extend beyond one's own lifespan. This phenomenon occurs through mechanisms such as the release of small RNA molecules, which can traverse the reproductive system and influence embryonic development. The implications of these findings highlight the potential for lifestyle choices to impact not just oneself, but one's offspring. The relationship between exercise and epigenetic modulation underscores a broader understanding of health. The environment, including factors such as physical activity, can significantly shape biological pathways. Research continues to explore how varying forms of exercise, endurance training, resistance training, and high-intensity interval training affect the epigenome in distinct ways. This area of study opens new avenues for enhancing public health strategies, emphasizing the importance of integrating physical activity into daily life as a means to promote long-term genetic health. Sleep plays a critical role in the regulation of epigenetic mechanisms, influencing gene expression and overall health. This state of rest allows the body to undergo vital restorative processes, which are essential for maintaining cellular function and resilience. During sleep, the brain engages in a series of complex activities, such as memory consolidation and waste clearance, that are mediated by epigenetic modifications. Though these modifications can be significantly influenced by the quality and duration of sleep, research suggests that insufficient sleep may lead to detrimental changes in the epigenome, which could have long-lasting effects on health outcomes. The interaction between sleep patterns and epigenetic changes highlights the intricate balance between lifestyle factors and genetic expression. Sleep deprivation, for instance, is associated with alterations in the expression of genes linked to stress response and metabolic regulation. This interplay also extends to the next generation, as the epigenetic impacts of sleep may be transmitted to offspring. The implications of this transgenerational effect underscore the importance of prioritizing restorative sleep, as it not only affects individual health, but may also shape the genetic predispositions of future generations. By understanding the science behind sleep and its epigenetic influences, strategies can be developed to improve sleep hygiene. The implications of epigenetic research extend beyond biology, raising important ethical considerations. This area of study reveals how environmental factors can influence not only individual health, but also the genetic predispositions of future generations. The intricacies of gene expression may lead to questions regarding parental responsibility and the potential burden of inherited traits. Informed consent becomes paramount in this context. Participants in epigenetic studies must clearly understand how their genetic information could impact their descendants. This region of research necessitates transparency about the potential risks and benefits, ensuring individuals are adequately informed prior to their involvement. The possibility of using epigenetic knowledge to manipulate genetic traits introduces additional complexities. As scientists explore targeted interventions, the line between therapeutic applications and enhancement blurs. Ethical debates arise surrounding issues of equity, access, and the implications of designer traits. Concerns about exacerbating social inequalities become increasingly relevant as the potential for gene editing technologies develops. Long-term consequences of epigenetic alterations require careful consideration. Changes in gene expression may have unforeseen effects, influencing not only health but also behavioral and psychological traits. These impacts can ripple through generations, necessitating a cautious approach to any applications emerging from this research. Balancing innovation with ethical standards remains essential in navigating the evolving landscape of epigenetic science. The future of epigenetic testing presents a landscape rich with potential and inherent challenges. This developing field promises insights into how lifestyle and environmental factors influence gene expression, offering a window into not only individual health, but also the well-being of future generations. Evidence suggests that understanding these epigenetic changes may allow for more personalized medical interventions, tailoring treatments based on an individual's unique genetic expression. Though the promise is significant, pitfalls remain. Issues of privacy and consent arise as genetic information becomes increasingly accessible. Questions emerge regarding who controls this data and how it may be utilized in decision-making processes, particularly in healthcare and insurance sectors. The complexity of epigenetic interactions complicates the interpretation of results, as environmental variables may obscure clear conclusions about health risks or benefits. The potential for misuse also looms dalexis as society grapples with the ethical implications of modifying epigenetic markers. Such actions could have unintended consequences, influencing not just the individual but their descendants as well. Education and public discourse will be critical in navigating these challenges. ensuring that advancements in epigenetic testing prioritize informed consent and ethical application. This careful approach aims to harness the promise of epigenetics while safeguarding against the pitfalls that may accompany its rapid evolution. Epigenetics offers a profound understanding of how individual experiences can impact genetic expression, influencing not only personal health but also that of future generations. This field reveals that environmental factors, lifestyle choices, and even emotional well-being can modify gene activity, creating changes that may persist across generations. The implications of this knowledge extend into the realm of personalized medicine, where treatments can be tailored to the unique epigenetic profiles of individuals. In this emerging paradigm, the integration of epigenetic data allows for a more nuanced understanding of diseases. Conditions previously thought to be solely hereditary may now be seen through the lens of environmental interactions. Personalized medicine utilizes this insight to develop targeted interventions, enhancing the efficacy of treatments. Genetic predispositions can be assessed in conjunction with epigenetic markers, providing a comprehensive view of health risks. This approach transcends traditional one-size-fits-all methodologies, emphasizing the importance of individual variability. By considering how lifestyle, diet, and stress levels influence gene expression, healthcare providers can craft strategies that resonate with each person's unique biological narrative. The potential for epigenetics to inform preventative measures and therapeutic responses is substantial, marking a significant shift toward more individualized healthcare solutions. Understanding these connections continues to refine the landscape of medicine, revealing the intricate interplay between genes and environment. Obesity represents a profound challenge, influenced by a complex web of genetic and environmental factors. Epigenetics plays a crucial role, demonstrating how lifestyle choices can modify gene expression without altering the underlying DNA sequence. In this context, the environment encompasses factors such as diet, physical activity, and stress. These elements can trigger epigenetic modifications, leading to changes in metabolism and fat storage. Research indicates that certain dietary patterns, particularly those high in sugar and saturated fats, can induce epigenetic changes that promote weight gain. These modifications can affect hormones that regulate appetite and energy balance, creating a cycle that perpetuates obesity. Additionally, the impact of maternal nutrition during pregnancy has been shown to influence the epigenetic landscape of the developing fetus, potentially predisposing future generations to obesity. The role of epigenetics extends beyond individual behaviors. Societal factors, including socioeconomic status and food accessibility, contribute to the epidemic of obesity. These influences can shape the epigenetic profile of populations, leading to disparities in health outcomes. Understanding these pathways illuminates potential interventions that could mitigate the effects of obesity, emphasizing the need for comprehensive strategies that address both genetic predispositions and environmental contexts. This exploration of epigenetics unveils a deeper understanding of weight management, highlighting the delicate balance between biology and behavior. All socioeconomic status significantly influences epigenetic profiles, revealing the intricate connections between environment and genetic expression. Access to resources such as nutrition, healthcare, and education plays a pivotal role in shaping these biological markers. Individuals from lower socioeconomic backgrounds often encounter higher stress levels, inadequate access to healthy food options, and limited healthcare, all contributing to distinct epigenetic alterations. Research indicates that adverse childhood experiences associated with socioeconomic disadvantage can lead to modifications in DNA methylation patterns. These changes may predispose individuals to various health conditions, including obesity, diabetes, and cardiovascular diseases. The cumulative effects of chronic stress and limited access to supportive environments can establish a cascade of biological responses, perpetuating a cycle of disadvantage across generations. Communities experiencing economic hardship frequently exhibit higher levels of environmental toxins, which further exacerbate epigenetic alterations. Exposure to pollutants can modify gene expression, influencing health outcomes in both the exposed individual and their offspring. Consequently, the interplay between socioeconomic factors and epigenetic changes underscores the importance of addressing systemic inequalities. Interventions aimed at improving socioeconomic conditions have the potential to modify these epigenetic landscapes by enhancing access to resources and supportive environments. It may be possible to foster healthier genetic profiles, promoting well-being across generations. This connection emphasizes the necessity of holistic approaches in public health initiatives. Family dynamics serve as a critical component in the intricate web of epigenetic programming. Interactions among family members can influence emotional well-being, behavioral patterns, and overall health, which in turn may leave lasting marks on genetic expression. This region of study highlights how stress, nurturing, and even the dynamics of conflict within a household can trigger epigenetic changes that transcend generations. Research has shown that children exposed to chronic stress or adverse experiences may exhibit alterations in gene expression linked to stress response. These modifications, although not changing the underlying DNA sequence, can affect how genes are turned on or off, potentially shaping the child's future health outcomes. Nurturing environments, characterized by supportive relationships, have been found to promote positive epigenetic changes, fostering resilience and reducing the risk of diseases. The interplay between genetic predisposition and family dynamics creates a unique landscape for understanding health. It underscores the importance of fostering environments that encourage healthy interactions and support. In this context, epigenetics becomes a window into how life experiences can influence not just individual health, but the broader familial lineage. Understanding this relationship invites a broader conversation about the implications for public health strategies. Encouraging positive family dynamics may be an essential step in promoting healthier future generations, making it a vital focus for research and intervention. Autodiox metically influence epigenetic mechanisms, shaping the biological outcomes for future generations through lifestyle choices and social environments. Traditions surrounding nutrition, physical activity, and stress management play pivotal roles in determining the health of individuals and their descendants. For instance, dietary customs rich in antioxidants and essential nutrients can alter gene expression, fostering resilience against chronic diseases. In contrast, diets high in processed foods may lead to detrimental epigenetic modifications, predisposing offspring to metabolic disorders. Social structures also contribute to the epigenetic landscape. Communities that prioritize mental well-being and support networks tend to cultivate environments that reduce stressors, promoting healthier gene expression patterns. Conversely, areas plagued by conflict or economic instability may exacerbate stress responses, creating negative epigenetic changes that can persist across generations. Educational practices further impact these dynamics, as access to knowledge about health and nutrition empowers individuals to make informed choices, potentially altering their genetic legacy. Parenting styles and child-rearing practices are equally critical, as nurturing environments can foster positive epigenetic outcomes, while neglect or abuse can lead to persistent epigenetic changes linked to psychological distress. Refondast these cultural implications illuminates pathways for health interventions that respect and integrate community values, ensuring that future generations may thrive in more favorable biological contexts. As education serves as a pivotal influence on epigenetic outcomes, shaping not only individual cognitive development but also broader societal health. Access to quality education fosters critical thinking skills, emotional regulation, and and resilience, all of which can have profound effects on gene expression. It empowers individuals to make informed choices regarding their health, nutrition, and lifestyle, contributing to a more favorable biological landscape for future generations. This region of study highlights the role of socioeconomic status in educational attainment. Disparities in educational opportunities can lead to significant differences in health outcomes, as those with limited access to education may experience increased stress, reduced health literacy, and diminished capacity to navigate health challenges. The implications of these factors extend beyond immediate health, influencing the epigenetic landscape of offspring. Research indicates that educated parents are more likely to engage in nurturing practices that promote positive developmental trajectories for their children. This nurturing environment facilitates healthy gene expression, which may mitigate the risks associated with adverse experiences. Consequently, educational initiatives that focus on equity and accessibility can create a ripple effect, enhancing community resilience and offering a pathway toward better health outcomes. Understanding the intricate connections between education, environment, and epigenetics underscores the importance of fostering equitable systems that nurture both individual potential and collective well-being. The Riften and Cethism via Bazirlux and evolutionary biology reveals profound insights into how organisms adapt over generations. This field explores how environmental factors can influence gene expression, allowing traits to be altered without changes to the underlying DNA sequence. Such modifications can be passed down through generations, providing a mechanism for rapid adaptation in response to shifting environments. Research indicates that these epigenetic changes can be triggered by various stimuli, including diet, stress, and exposure to toxins. This adaptability highlights a dynamic interplay between genetic inheritance and environmental pressures. In this context, evolutionary biologists examine how these changes impact survival and reproductive success, providing a more nuanced understanding of natural selection. In diverse ecosystems, populations exhibiting advantageous epigenetic traits may thrive while others decline, shaping the evolutionary trajectory of species. The implications extend to understanding human health and disease, where epigenetic factors can influence susceptibility to conditions such as obesity and diabetes. These insights suggest that evolution is not solely a matter of gradual genetic shifts, but also a complex interplay of environmental influences and heritable modifications. Further exploration of this relationship may illuminate the pathways through which species navigate their environments, adapting not only genetically, but also epigenetically, thereby enriching the broader narrative of life's persistence and resilience. Cross-species comparisons reveal fascinating epigenetic mechanisms at play within various animal populations. In mammals, for example, maternal care has been shown to induce lasting epigenetic changes in offspring. The nurturing behaviors exhibited by mothers can influence the expression of genes associated with stress response, effectively altering their young's physiological and behavioral traits. Birds present another intriguing case. In certain species, environmental factors like habitat variability and food availability lead to epigenetic adjustments that affect song patterns, showcasing a direct link between surroundings and genetic expression. These adaptations can enhance reproductive success, demonstrating how epigenetics facilitates real-time responses to ecological demands. Reptiles illustrate yet another dimension of this complexity, where temperature-dependent sex determination results from epigenetic mechanisms. Fluctuations in temperature during critical developmental stages can dictate whether hatchlings emerge as male or female, underscoring the profound influence of environmental conditions on genetic expression. In aquatic environments, fish exhibit remarkable adaptability through epigenetic changes in response to pollution and habitat degradation. These alterations can affect metabolic pathways and reproductive strategies, enabling populations to withstand adverse conditions. Such cross-species comparisons underscore the ubiquity of epigenetic mechanisms across the animal kingdom, illustrating a dynamic interplay between genetics and environmental contexts. This multidimensional perspective enriches the understanding of evolutionary processes, revealing the intricate ways life persists and evolves within diverse ecosystems. The role of epigenetics in plant biology emerges as a crucial factor in understanding adaptation and resilience. Plants, often subjected to environmental stresses such as drought, salinity, and temperature fluctuations, exhibit remarkable capacity to modify their gene expression in response to external stimuli. This flexibility enables them to survive and thrive in varying conditions, ensuring their continued presence in diverse habitats. These mechanisms involve chemical modifications that influence gene activity without altering the underlying DNA sequence. Methylation and histone modification are two key processes through which plants can activate or silence specific genes based on environmental cues. Such adaptations not only enhance individual survival, but can also impact the genetic landscape of entire populations over generations. In agriculture, harnessing epigenetic principles can lead to the development of more resilient crop varieties. Breeding programs that incorporate epigenetic insights may produce plants capable of withstanding climate challenges, thus contributing to food security. Researchers are increasingly investigating how epigenetic markers can be used to select for desirable traits, such as improved yield or nutritional quality. Sulpernomaly. UMSALS reveals a deeper connection between environmental factors and genetic expression in the plant kingdom. The implications of this understanding extend beyond individual species, influencing agricultural practices and ecosystem management in a rapidly changing world. Epigenetic changes in response to climate change reveal a complex interplay between environmental factors and genetic expression. Organisms adapt to shifting climates through modifications that can influence their survival and reproductive success. This region, characterized by fluctuating temperatures and altered precipitation patterns, compels species to adjust their physiological responses. These adjustments often manifest as changes in gene expression rather than alterations in the underlying DNA sequence. The capacity of certain plants to modify their growth patterns in response to drought conditions illustrates this adaptability. Through epigenetic mechanisms, such as DNA methylation and histone modification, these organisms can enhance their resilience. This resilience is not merely a temporary adjustment. It can be passed on to subsequent generations. Such transgenerational effects indicate that environmental pressures shape not just the immediate population, but also future cohorts. In animal populations, these epigenetic responses can influence behavior, reproductive timing, and stress responses. The ability to adjust to environmental stresses without genetic changes allows for a more rapid response to climate fluctuations. This phenomenon is observable in various ecosystems, where species must navigate the dual challenges of habitat loss and increased competition. Understanding these epigenetic dynamics provides insight into the resilience of life forms amidst a shifting climate, highlighting the intricate connections between genetics, environment, and survival. Advancements in technology have significantly enhanced the study of epigenetics, allowing researchers to delve deeper into the mechanisms that govern gene expression. High-throughput sequencing techniques enable the analysis of DNA methylation and histone modifications with unprecedented precision. This capability offers a clearer view of how environmental factors can influence genetic outcomes across generations. Microarrays and CRISPR-Cas9 gene editing are pivotal tools in this exploration. facilitating targeted investigations into specific epigenetic changes. These instruments allow scientists to manipulate and observe particular genes, revealing the nuanced ways in which external stimuli, such as diet and stress, can alter epigenetic patterns. Techniques like RNA sequencing further illuminate the functional consequences of these modifications, linking changes in gene expression to observable traits. Data analytics and computational biology play critical roles, processing vast amounts of information generated by these technologies. Machine learning algorithms analyze complex datasets, identifying patterns and correlations that might remain hidden through traditional methods. This integration of computational power enhances the understanding of epigenetic phenomena, paving the way for potential therapeutic interventions. In clinical settings, epigenetic biomarkers are being developed to predict disease risk and treatment efficacy, underscoring the practical applications of this research. The convergence of technology and epigenetics holds promise for addressing health disparities and advancing personalized medicine, offering a glimpse into a future where genetic predisposition can be actively managed. The epigenetic clock represents a groundbreaking approach to understanding biological age beyond chronological markers. This innovative tool relies on the measurement of DNA methylation patterns, which change as an organism ages. These modifications provide insights into the cellular aging process, revealing how lifestyle factors and environmental exposures impact longevity. Researchers have identified specific sites on the genome that correlate with age, allowing them to create algorithms that predict biological age with remarkable accuracy. This region of study has opened avenues for understanding diseases linked to aging, such as Alzheimer's and cardiovascular conditions. By assessing how epigenetic changes may signal early onset, the implications of the epigenetic clock extend beyond mere observation. It offers potential interventions to slow down or even reverse aspects of aging. Lifestyle modifications, dietary changes, and stress management can all influence DNA methylation, effectively recalibrating the clock. In clinical settings, this tool could enhance preventive medicine, enabling healthcare providers to tailor interventions based on an individual's biological age rather than chronological age. Emerging research continues to explore the intricate relationship between genetic expression and environmental factors, emphasizing the dynamic nature of the human genome. This evolving understanding challenges traditional views of heredity, paving the way for a more nuanced approach to health and wellness across generations. Quantifying epigenetic changes involves various methodologies designed to analyze modifications to the genome. Techniques such as bisulfite sequencing provide insights into DNA methylation patterns, revealing how environmental factors can influence gene expression. This process allows researchers to observe changes at a granular level, unveiling the interplay between genetics and lifestyle. Chromatin immunoprecipitation combined with sequencing, commonly known as ChIP-seq, further complements these findings. This method identifies protein-DNA interactions, shedding light on how epigenetic markers regulate transcription. By focusing on the binding sites of transcription factors and histones, or DORs, it becomes possible to discern the specific regions within the genome that respond to external stimuli. Microarray technology offers another perspective, enabling the simultaneous analysis of thousands of gene expressions. This technique measures RNA levels, providing a snapshot of gene activity under various circumstances. Such data can reveal how stress, nutrition, and exposure to toxins alter the epigenetic landscape. Recent advancements in single-cell sequencing allow for the examination of epigenetic changes at an unprecedented resolution. By analyzing individual cells, researchers can uncover heterogeneity in epigenetic modifications, highlighting unique responses to environmental influences. Together, these methodologies create a comprehensive framework for understanding how epigenetic changes manifest, emphasizing the intricate relationship between inherited traits and external factors shaping health across generations. The intricate mechanisms of RNA play a pivotal role in regulating epigenetic processes. Various types of RNA, including small interfering RNA and long non-coding RNA, influence gene expression without altering the underlying DNA sequence. This regulation occurs primarily through interactions with chromatin, the complex of DNA and proteins that forms chromosomes. These RNA molecules can recruit or inhibit the activity of enzymes responsible for adding or removing epigenetic marks. Particularly, small interfering RNA acts by directing the silencing of specific genes, leading to a dynamic adjustment of gene expression in response to environmental stimuli. Long non-coding RNA, on the other hand, often serves as scaffolds that organize chromatin-modifying proteins, effectively altering the structural configuration of chromatin. This alteration determines whether certain genes are accessible for transcription or remain dormant. In addition, RNA molecules participate in the establishment of epigenetic memory, allowing organisms to retain a record of environmental exposures. This memory can be passed down through generations, reinforcing the notion that phenotypic changes are not solely the result of genetic inheritance. The interplay between RNA and epigenetic regulation underscores the complexity of heredity, illustrating how life experiences can imprint lasting modifications that affect descendants. Such insights hold significant implications for understanding developmental biology and potential therapeutic avenues for genetic disorders. Histone modifications serve as a crucial aspect of the epigenomic landscape, influencing gene expression without altering the underlying DNA sequence. These chemical changes can be likened to markers that guide the accessibility and transcriptional activity of genes. Specific enzymes add or remove these modifications, creating a dynamic regulatory environment that responds to various physiological stimuli. Acetylation, methylation, and phosphorylation represent some of the key modifications that occur on histone proteins. Acetylation typically relaxes chromatin structure, facilitating gene expression, while methylation can either activate or repress transcription, depending on the context. This dual role highlights the complexity of the regulatory mechanisms at play. Cells utilize these modifications to adapt to diverse environmental factors, including stress and nutrition, effectively transmitting information to subsequent generations. The stability of histone marks ensures that certain epigenetic states can persist through cell division, impacting developmental trajectories and potentially influencing phenotypic outcomes in descendants. Research continues to uncover the intricate patterns and combinations of histone modifications that form a code for gene regulation. Understanding this language of the epigenome not only enriches our grasp of cellular processes, but also opens avenues for innovative strategies in addressing genetic disorders. It becomes evident that every experience might leave an imprint, shaping not just individual lives, but also the genetic legacies passed to future generations. The fultus sulfat the faulted morsel, the tinterceltra the falter fultus. This region, mechanisms of gene regulation, reveal a profound complexity, particularly through the role of non-coding RNAs. These molecules, once dismissed as mere genomic noise, are now recognized as critical players in the fine-tuning of genetic expression. They operate in various forms, including microRNAs and long non-coding RNAs, each contributing uniquely to cellular functions and developmental processes. MicroRNAs, for instance, engage in the precise modulation of messenger RNA, influencing the production of proteins essential for growth and development. This regulatory capacity allows for a dynamic response to environmental cues, ensuring that cellular activities align with external demands. The impact of these small molecules extends beyond mere regulation. They are involved in processes such as differentiation, proliferation, and apoptosis, shaping the very fabric of life. Long non-coding RNAs introduce another layer of complexity, interacting with chromatin and transcription factors to orchestrate gene expression at multiple levels. Their presence signifies a regulatory network that transcends traditional paradigms of genetic control. This network highlights the sophistication of biological systems, where non-coding regions of the genome play pivotal roles in determining the fate of cells and, subsequently, the organism as a whole. In this context, the epigenetic landscape is further enriched, revealing pathways through which life experiences may influence not only individual health, but also the genetic composition of descendants. Maternal nutrition serves as a crucial factor in shaping the epigenetic landscape of offspring. Nutritional intake during pregnancy can lead to modifications in the DNA methylation patterns of the developing fetus. These alterations have lasting implications, potentially influencing gene expression and overall health outcomes for the child. Specific nutrients, such as folate, choline, and omega-3 fatty acids, are particularly significant. They participate in crucial biochemical pathways, affecting the establishment of epigenetic marks that regulate various developmental processes. In this region, evidence suggests that a balanced maternal diet may promote optimal growth and reduce the risk of chronic diseases later in life. Conversely, deficiencies or imbalances in nutrition can predispose offspring to metabolic disorders, obesity, and cardiovascular disease. The timing and nature of nutrient availability are fundamental, as certain critical windows during gestation may amplify the impact of maternal nutrition on epigenetic programming. These findings underscore the intricate relationship between environmental factors and genetic expression. They also raise essential considerations for public health, emphasizing the need for nutritional guidance for expectant mothers. The intergenerational transmission of epigenetic modifications highlights a pathway through which lifestyle choices can resonate through subsequent generations, illustrating the profound connections between biology, environment, and health. Understanding this dynamic presents opportunities for targeted interventions aimed at enhancing maternal and child health outcomes. Substance abuse introduces significant alterations to the epigenetic landscape. Exposure to addictive substances can lead to modifications in DNA methylation and histone modifications, which may result in lasting changes not only for the individual, but also for their offspring. These changes can influence gene expression in ways that affect physical health, behavior, and susceptibility to addiction. Research indicates that maternal substance use during pregnancy can have profound implications for fetal development. Chemicals from substances such as alcohol, nicotine, and opioids cross the placental barrier, altering the epigenetic mechanisms that govern growth and neurological development. This interference can lead to developmental disorders and an increased risk of behavioral issues in children. As recordings, we dixates, the transgenerational effects must be considered. Offspring may inherit not only the behavioral tendencies of their parents, but also the altered gene expressions resulting from substance exposure. Consequently, individuals raised in environments where substance abuse is prevalent may face compounded risks as they navigate their own health and well-being. Understanding these epigenetic outcomes reinforces the importance of early intervention and support systems. Addressing maternal health and providing resources can mitigate the risks associated with substance abuse, promoting healthier futures for both mothers and children. These insights can guide public health initiatives aimed at breaking the cycle of addiction and fostering resilience in future generations. Inherited diseases present a compelling landscape for examining epigenetics. Specific conditions, such as cystic fibrosis and sickle cell anemia, have long been studied through a genetic lens. Recent findings suggest that epigenetic modifications may also play a crucial role in their manifestation. In cystic fibrosis, for instance, the presence of a mutated gene typically leads to severe respiratory complications. However, the variability in symptom severity among individuals can be partially explained by epigenetic factors that influence gene expression. Sickle cell anemia serves as another poignant example. This disease arises from a single nucleotide mutation. Nevertheless, epigenetic regulation may determine the extent of symptoms experienced by affected individuals. Certain epigenetic markers can silence or activate genes responsible for producing hemoglobin, thereby affecting the severity of the disease. Research has revealed that environmental factors, such as diet and stress, can induce epigenetic changes that may be passed down through generations. This phenomenon underscores the importance of understanding not only the genetic code inherited from parents, but also the epigenetic modifications that shape the health profiles of offspring. Animal studies further illuminate these concepts, showcasing how ancestral experiences can leave lasting marks on the genome, influencing the health of subsequent generations. Through these case studies, the intricate interplay between genetics and epigenetics reveals pathways for potential therapeutic interventions. The relationship between epigenetics and immunity demonstrates the profound impact of environmental factors on immune responses. Various stimuli, including pathogens and dietary elements, can cause modifications to the epigenome. Such changes influence gene expression patterns. Research indicates that certain epigenetic markers are associated with immune-related diseases. For instance, specific modifications can increase susceptibility to autoimmune disorders, where the immune system mistakenly attacks the body's own tissues. These markers can serve as indicators of risk, guiding future medical interventions and preventive strategies. In addition to disease susceptibility, epigenetics plays a crucial role in the adaptability of the immune system. During infections, it can regulate the expression of genes responsible for producing antibodies and activating immune cells. The dynamic nature of this regulation allows the immune system to respond effectively to new threats. Understanding this connection between epigenetics and immunity offers valuable insights into how lifestyle choices can lead to significant health outcomes. The implications extend beyond individual health, suggesting that the effects of epigenetic changes may echo across generations, shaping the immune resilience of descendants. Understanding epigenetics in stem cell biology opens avenues for innovations in regenerative medicine and therapeutic strategies. Stem cells possess the unique ability to differentiate into various cell types, a process influenced not only by genetic factors, but also by epigenetic modifications. These alterations can determine the fate of a stem cell, guiding it towards becoming a neuron, muscle cell, or any other specialized cell type. Research demonstrates that environmental factors, such as nutrition and exposure to toxins, can modify the epigenetic state of stem cells. Such modifications could improve or hinder their ability to regenerate tissues. This region of study is particularly promising for treating degenerative diseases, where restoring function to damaged tissues is essential. By understanding how epigenetic changes affect stem cell behavior, scientists can develop strategies to manipulate these mechanisms, potentially enhancing the efficacy of stem cell therapies. Clinical applications may include the reprogramming of adult stem cells to treat conditions like spinal cord injuries or neurodegenerative diseases. Furthermore, the potential to reverse the epigenetic marks that lead to disease offers a unique approach to personalized medicine. This knowledge lays the groundwork for targeted interventions that could not only repair existing damage, but also prevent future health issues by enhancing stem cell resilience and adaptability. The influence of epigenetics extends profoundly into fertility and reproductive health. In this domain, it unveils the intricate connections between environmental factors, parental health, and the genetic blueprint passed to future generations. Alterations in DNA methylation and histone modification can significantly affect gamete quality. These biochemical changes, influenced by lifestyle, nutrition, and stress, can shape the viability of sperm and eggs. This region of study emphasizes that both maternal and paternal epigenetic modifications may impact conception outcomes. For instance, stresses experienced by parents can lead to epigenetic changes that may disrupt normal embryonic development, potentially resulting in complications. The epigenetic landscape of gametes can determine not only successful fertilization, but also subsequent implantation and fetal growth. Research indicates that the maternal environment, including factors such as diet and exposure to toxins, can influence epigenetic markers in the developing embryo. These markers can persist, affecting health and development throughout life. Conversely, paternal contributions are equally significant. Emerging data suggests that alterations in sperm epigenetics can influence offspring metabolic health and susceptibility to diseases. Understanding these dynamics offers insights into reproductive health strategies, emphasizing the need for comprehensive preconception care. This knowledge underscores the importance of holistic approaches to fertility, considering the broader implications of epigenetic research on future generations. Epigenetic regulation plays a pivotal role in the processes of learning and memory. This mechanism influences gene expression without altering the underlying DNA sequence, allowing for dynamic responses to environmental stimuli and experiences. In this context, certain genes become activated or silenced based on interactions with the external environment, shaping cognitive abilities over time. Research has shown that experiences such as stress, education, and social interactions can lead to epigenetic modifications in the brain. These alterations can enhance synaptic plasticity, a crucial feature for forming and retaining memories. In this region, specific proteins are involved in the remodeling of synapses, facilitating the integration of new information. The hippocampus, a key area associated with learning and memory, shows significant epigenetic activity. When individuals engage in learning, the expression of genes associated with neural growth and connectivity is upregulated. Conversely, adverse experiences may inhibit these processes, leading to deficits in cognitive function. Such findings highlight the intricate relationship between environment and biology, illustrating how life experiences can leave a lasting mark on the genetic framework of the brain. Genetic expression in response to environmental factors can inform strategies for enhancing cognitive health and resilience, emphasizing the potential for interventions that can modify epigenetic effects positively. Chronic diseases manifest as complex interactions between genetic predispositions and environmental influences. Epigenetics plays a pivotal role in this dynamic, as it provides a framework for understanding how lifestyle choices and exposures can modify gene expression related to these conditions. This region of study reveals that factors such as diet, Stress and pollution not only affect individuals, but can also have cascading effects on their descendants. Research indicates that the epigenetic modifications can lead to the activation or silencing of genes associated with inflammation, metabolism, and cellular repair. These changes often contribute to the development of conditions such as diabetes, cardiovascular disease, and various forms of cancer. By examining the molecular underpinnings of these processes, it becomes clear that a systems approach is essential. This perspective considers the intricate web of gene-environment interactions, allowing for a more comprehensive understanding of disease etiology. Interventions designed to target these epigenetic markers can potentially reverse or mitigate the effects of chronic diseases. Nutritional adjustments, physical activity, and stress management strategies may lead to favorable epigenetic changes, enhancing overall health. Investigating these connections underscores the importance of integrative health practices, highlighting the necessity of a holistic approach to prevent and treat chronic diseases effectively. Such insights pave the way for personalized medicine, where interventions are tailored to individual epigenetic profiles. Global perspectives on epigenetic research and policy reveal a landscape rich with diversity. Nations around the world are initiating studies aimed at understanding how environmental factors influence genetic expression. In Europe, collaborative efforts among countries focus on the implications of epigenetic changes in public health, advocating for policies that incorporate these findings into healthcare systems. This region is increasingly recognizing the importance of integrating epigenetic data into preventive medicine. In North America, research institutions are leading investigations into the long-term effects of lifestyle choices such as diet and stress on genetic markers. These studies emphasize the need for guidelines that promote healthy living with the potential to enhance overall societal well-being. The ethical considerations surrounding these findings prompt discussions about genetic privacy and the potential for discrimination based on epigenetic information. Epigenetic presents a unique perspective, with rapid advancements in technology allowing for extensive research into epigenetic modifications. Countries in this region are focusing on agricultural applications, exploring how epigenetics can influence crop resilience and yield. This could lead to sustainable practices that address food security. In Africa, the focus is on understanding how socioeconomic factors impact epigenetic health, aiming to improve health outcomes in underserved populations. This complex global dialogue continues to evolve, reflecting the intricate interplay between genetics, environment, and policy. Emerging directions in epigenetic research are poised to unveil deeper insights into how environmental factors influence genetic expression across generations. This growing field seeks to elucidate the mechanisms by which lifestyle choices and environmental exposures can impart lasting effects on DNA. Investigators are delving into the role of nutrition, stress, and even social dynamics in shaping epigenetic landscapes, revealing how these influences may alter gene expression and consequently health outcomes. Innovative technologies are accelerating this exploration. Advanced sequencing techniques allow for a more precise mapping of epigenetic modifications, offering a clearer picture of how genes are regulated. The integration of big data analytics enhances the ability to identify patterns and correlations, leading to more robust conclusions about the connections between environment and epigenetics. Additionally, the potential for therapeutic interventions arises from this understanding. These strategies aim to reverse detrimental epigenetic changes, restoring healthy gene function. Research into pharmacological agents that target epigenetic modifications is expanding, providing hope for conditions once deemed untreatable. Ethical considerations accompany these advancements, The implications of modifying epigenetic expressions raise questions about the long-term effects on future generations and the societal impact of such interventions. As this field progresses, it navigates a delicate balance between scientific innovation and moral responsibility, charting the course toward a deeper understanding of life's complexities. Public perception of epigenetics reflects a complex interplay of curiosity, skepticism, and fascination. Individuals often find themselves intrigued by the idea that experiences and environmental influences can affect not just personal health, but also the genetic legacy passed to future generations. This notion challenges long-held beliefs about heredity, prompting a re-evaluation of how one perceives familial traits and health predispositions. The implications of epigenetic research extend beyond scientific circles, influencing discussions in education, healthcare, and personal choices. Many individuals seek to understand how lifestyle modifications, such as diet, exercise, and stress management, can lead to changes in their epigenetic markers. This evolving dialogue fosters a sense of empowerment as individuals consider the potential to influence their health and that of their offspring. Yet, misunderstandings abound. Misconceptions about epigenetics can lead to the oversimplification of complex biological processes. Some may erroneously believe that all traits are malleable, ignoring the intricate interplay of genetics and environment. Misinformation may proliferate, particularly through social media platforms, complicating the public's grasp of scientific realities. Additionally, ethical concerns regarding manipulation of epigenetic factors provoke significant debate. The possibilities of enhancing human capabilities or altering genetic expressions raise questions about the moral implications of such interventions. As this field evolves, fostering an informed public discourse becomes essential for navigating these intricate themes. Non-governmental organizations play a vital role in enhancing awareness about epigenetics and its implications for public health. These entities engage in educational initiatives designed to bridge the gap between scientific research and community understanding. By offering workshops, seminars, and outreach programs, they disseminate knowledge on how lifestyle choices impact genetic expression across generations. This commitment to education fosters a more informed populace, encouraging individuals to take proactive steps toward healthier living. Campaigns emphasizing nutrition, physical activity, and stress management are common strategies employed to illustrate the connection between environment and genetics. Such efforts highlight the significance of epigenetic factors in chronic disease prevention and overall well-being. In addition to education, these organizations often advocate for policy changes that reflect emerging scientific insights. They work diligently to influence legislation regarding environmental protection, healthcare access, and nutritional guidelines. By aligning their objectives with scientific findings, they contribute to a more comprehensive approach to public health. Collaboration with researchers enhances the credibility of their initiatives, ensuring that the information disseminated is grounded in rigorous scientific inquiry. This partnership enables the translation of complex data into accessible formats, allowing for broader public engagement. Through these multifaceted efforts, non-governmental organizations cultivate a deeper understanding of epigenetics, underscoring its relevance in contemporary discourse. This relevance extends into public health, where the implications of epigenetic research shape strategies for implementation. Community health initiatives increasingly incorporate findings on how environmental factors, lifestyle choices, and stressors influence genetic expression. These programs emphasize education about nutrition, mental well-being, and toxin exposure. The goal is to foster healthier environments that can mitigate adverse epigenetic changes across generations. Public health officials utilize data-driven approaches to tailor interventions that resonate with diverse populations. These strategies are built on a foundation of scientific understanding that highlights the interplay between genetics and environment. They aim to empower individuals with knowledge that promotes informed lifestyle decisions, potentially altering the trajectory of hereditary health issues. Collaboration between researchers and public health entities enhances outreach efforts, ensuring that epigenetic concepts are accessible and applicable. Workshops, seminars, and digital resources serve as vehicles for disseminating information, creating a network of informed communities. The impact of such initiatives may be profound, as they strive to break cycles of disease that have persisted through genetic predisposition. By focusing on the modifiable aspects of health influenced by epigenetics, there lies the potential to cultivate resilience within populations, thereby fostering a healthier future for subsequent generations. The essence of this approach lies in its commitment to bridging science with practical application. Understanding the intricate layers of epigenetic landscapes reveals a conceptual framework that underpins how environmental factors influence genetic expression across generations. This phenomenon occurs through chemical modifications to DNA and the proteins associated with it, affecting how genes are turned on or off without altering the underlying genetic sequence itself. Such modifications can be triggered by a range of influences, including diet, stress, and exposure to toxins. This region of study emphasizes the dynamic nature of genes, showing that they are not static blueprints but rather responsive entities shaped by external conditions. The interplay between genetic predisposition and epigenetic changes creates a complex network of interactions that can manifest in observable traits and health outcomes. Stresses encountered by one generation may imprint changes that echo into the next, effectively encoding experiences into the very fabric of DNA. In this framework, the role of nutrition emerges as pivotal, with specific nutrients capable of modifying epigenetic markers, thereby influencing overall health trajectories. These modifications can persist, potentially affecting not only immediate offspring, but also extended generations, thereby altering the course of familial health histories. Understanding this nuanced landscape offers insights into disease prevention, emphasizing the importance of environmental factors in shaping genetic legacies. The interplay between genetics and epigenetics reveals a complex relationship that shapes individual traits and health outcomes. Genetic codes, composed of sequences that determine inherited characteristics, serve as the foundational blueprint. Epigenetic modifications, on the other hand, act as regulators, influencing how these genetic instructions are expressed without altering the underlying DNA sequence. This dual approach provides a more comprehensive understanding of biological development and potential responses to environmental stimuli. Mechanisms such as DNA methylation and histone modification operate within this intricate framework, dynamically adjusting gene expression in response to various external factors. These changes can be transient or stable, potentially lasting across generations. The impact of lifestyle choices, nutritional habits, and stress exposure is profound, as they can induce epigenetic changes that influence not only the individual but also their descendants. Research indicates that this interaction plays a critical role in the onset of certain diseases, illustrating how inherited vulnerabilities may be exacerbated or mitigated by environmental conditions. In this region of study, the implications extend beyond simple inheritance patterns, revealing a continuum of influence that can shape populations over time. The understanding of this duality offers essential insights into the mechanisms of adaptation and resilience, paving the way for targeted interventions in public health and personalized medicine. Epigenetics plays a crucial role in shaping the microbiome, reflecting the intricate interplay between genetics and environmental factors. This relationship illustrates how external influences can modify gene expression without altering the DNA sequence itself. Microbial communities residing within the human body can impact these epigenetic changes, demonstrating a feedback loop that enhances or diminishes certain traits. Diverse microbial populations contribute to an individual's health by influencing metabolic pathways, immune responses, and even behavior. They interact with host cells, releasing metabolites that can signal changes in gene expression. This dynamic interaction highlights the importance of maintaining a balanced microbiome, as disruptions can lead to epigenetic alterations associated with various diseases. The influence of diet on both the microbiome and epigenetic modifications is significant. Nutrient intake can shape microbial diversity, while also providing the necessary substrates for epigenetic processes. For instance, compounds such as folate and other B vitamins can affect DNA methylation patterns, potentially impacting future generations. Research continues to unveil the complexities of this symbiotic relationship. Understanding it not only advances knowledge in health and disease management, but also emphasizes the importance of lifestyle choices. This connection between lifestyle choices and epigenetic impact has brought technology to the forefront of research. Advanced sequencing techniques enable scientists to delve deeper into the modifications that occur within DNA, revealing how external factors can lead to lasting changes. High-throughput sequencing has accelerated the identification of epigenetic markers, providing insights into how environmental influences can alter gene expression without changing the underlying genetic code. CRISPR technology has emerged as a pivotal tool in the manipulation of epigenetic states. By precisely targeting specific genes, researchers can introduce or remove epigenetic modifications. This capability opens avenues for therapeutic interventions, potentially reversing detrimental epigenetic alterations associated with various diseases. Techniques such as RNA interference further complement this arsenal, allowing for the silencing or activation of genes based on epigenetic signals. The integration of artificial intelligence enhances the ability to analyze vast datasets generated from epigenomic studies. Machine learning algorithms can identify patterns and predict outcomes, facilitating the development of personalized medicine strategies. These advancements underscore the potential for targeted therapies that consider individual epigenetic landscapes. As research progresses, the ethical implications of such technologies will necessitate careful consideration. The capacity to manipulate epigenetics raises fundamental questions about the extent of human intervention in our biological inheritance. These discussions will shape the future trajectory of epigenetic research. Ethical dilemmas arise prominently in the context of gene editing and epigenetic research. The potential to alter genetic information raises questions about the implications of such interventions on future generations. Uncertainty looms over the long-term consequences of modifying DNA, particularly when considering traits that may extend beyond immediate health benefits. Ethical frameworks must navigate the murky waters of consent, especially when the subjects are yet to be born. This region of inquiry highlights the delicate balance between scientific advancement and moral responsibility. The prospect of eradicating genetic diseases can be enticing, yet it may inadvertently usher in unforeseen complications. The idea of enhancing human traits, such as intelligence or physical ability, introduces a new layer of complexity, as societal inequalities may be exacerbated by access to such technologies. Public sentiment plays a crucial role in shaping the discourse surrounding these advancements. Debates often center around the distinction between therapeutic interventions and enhancements. This distinction can determine the ethical acceptability of various practices. As researchers navigate this landscape, collaboration with ethicists, policymakers, and the public becomes essential. Clear guidelines and regulations will be necessary to ensure responsible usage of these powerful tools. The landscape of epigenetics and gene editing calls for a thoughtful approach, considering both scientific potential and ethical ramifications. Cascading effects of globalization extend into the realm of epigenetic profiles, influencing gene expressions within local populations. This phenomenon occurs as cultural exchanges reshape dietary habits, lifestyles, and environmental exposures. These factors interact intricately with genetic material, leading to observable changes in health and disease susceptibility across generations. Local communities experience shifts in their epigenetic landscapes through increased contact with diverse populations, introducing new environmental stressors and nutritional elements. It is observed that urbanization, often a byproduct of globalization, alters exposure to pollutants and modifies physical activity patterns, all of which can induce epigenetic modifications. Studies indicate that migration patterns contribute to these changes as individuals adapt to new climates and dietary resources. Such adaptations may lead to differential gene expression responses that can be inherited, creating a mosaic of epigenetic variations within a community. The implications of these alterations are profound, suggesting that the choices made on a global scale reverberate down to the microscopic level of individual DNA. Each adaptation carries with it the potential for both beneficial and adverse health outcomes, making the understanding of these influences essential for public health initiatives. Balancing benefits of globalization with the preservation of local health traditions remains a critical challenge as these processes continue to evolve and intertwine. This region of inquiry has witnessed a notable shift with the advent of citizen science, which invites individuals outside of professional research environments to contribute to data collection and analysis. It fosters a unique collaboration between laypersons and scientists, enhancing the breadth of research through diverse perspectives and localized knowledge. Individuals participating in these initiatives often provide insights that may not be captured through traditional research methodologies. Data gathered by citizen scientists can reveal variations in epigenetic markers influenced by environmental factors, lifestyle choices, and dietary habits. This information significantly contributes to understanding how external conditions can lead to changes in gene expression across generations. Such contributions may bridge gaps in knowledge regarding epigenetic mechanisms, allowing for a more nuanced understanding of health and disease. Engagement in these studies empowers communities, creating a sense of ownership over health outcomes. Participants become co-creators of knowledge, influencing research directions and priorities. This grassroots approach not only democratizes science but also facilitates the integration of local health practices within broader epigenetic research frameworks. Innovations in epigenetic research are poised to redefine the landscape of genetic understanding. Emerging technologies, such as CRISPR-Cas9 and advanced sequencing methods, enable precise manipulation and analysis of epigenetic modifications. These developments hold the potential to unravel the intricacies of how environmental factors influence gene expression across generations. In this evolving field, researchers are increasingly focusing on the role of the microbiome and its influence on epigenetic changes. The complex interactions between gut health and gene regulation suggest that dietary choices and lifestyle factors may have profound implications for epigenetic inheritance. This relationship may lead to novel preventative strategies for diseases that have both genetic and environmental components. Collaboration between disciplines is essential in advancing knowledge. Integrating insights from psychology, sociology, and environmental science can illuminate the multifaceted nature of epigenetic influences. These interdisciplinary approaches may uncover how stress, socioeconomic status, and exposure to pollutants impact gene expression and health outcomes. Longitudinal studies will become vital in tracking epigenetic changes over time by observing individuals across their lifespan. Researchers can better understand the dynamic nature of epigenetics and its implications for future generations. The integration of big data analytics will further enhance the ability to identify patterns and correlations, paving the way for targeted interventions in public health. Interdisciplinary approaches to studying epigenetics reveal the complex interactions between environmental factors and genetic expression. This field draws from areas such as molecular biology, genetics, psychology, and even sociology, forming a multifaceted understanding of how experiences shape DNA. Researchers utilize advanced imaging techniques to visualize cellular changes, allowing them to observe how external stimuli influence gene activity over time. By employing bioinformatics, they analyze vast datasets that encompass genetic information alongside lifestyle variables. These analyses shed light on the influence of stress, nutrition, and exposure to toxins on gene regulation. Such insights emphasize that genetics is not solely a predetermined blueprint. It is a dynamic entity, intricately influenced by the surrounding environment. Collaborative efforts between geneticists and psychologists help decipher the mechanisms by which parental experiences can affect offspring. This relationship showcases the potential for behavioral traits to be passed down, not through direct genetic inheritance, but through epigenetic modifications. Environmental factors can lead to methylation changes, altering gene expression without changing the underlying DNA sequence. Understanding these connections fosters a deeper appreciation for the influence of societal conditions on health outcomes. It underscores the importance of addressing social determinants of health, which can have far-reaching implications for future generations. Case studies in epigenetic research reveal profound insights into how environmental factors can alter gene expression and influence subsequent generations. One study examined the effects of famine on the offspring of individuals who experienced severe nutritional deprivation. The findings indicated that the descendants of those who endured such hardships exhibited changes in metabolic responses, indicating a lasting impact on health profiles. This phenomenon illustrates the capacity of external conditions to instill alterations that transcend individual experiences. Another investigation considered the influence of stress on gene regulation, particularly in communities facing chronic adversity. It was observed that prolonged exposure to stressors resulted in epigenetic modifications that affected hormonal responses in the next generation. These changes can predispose descendants to various health challenges, reiterating the interconnectedness of life experiences and biological outcomes. Research focusing on maternal care in animal models further supports these concepts. Variations in maternal behaviors were linked to differences in gene expression in offspring, highlighting the significance of nurturing environments. These findings emphasize the intricate relationship between behavior, environment, and genetic expression, revealing that parental actions can leave an indelible mark on future generations. The implications of such studies extend beyond academic inquiry, guiding public health initiatives aimed at mitigating adverse social factors and fostering healthier communities. Understanding these dynamics is crucial for developing comprehensive strategies to enhance population well-being. The societal implications of epigenetic research extend beyond individual health. influencing public policy and educational framework. This field uncovers how environmental factors, lifestyle choices, and social conditions can affect genetic expression. By impacting future generations, it highlights the importance of addressing disparities in access to health resources, education, and nutrition, as these factors can fundamentally alter the epigenetic landscape. Communities grappling with socioeconomic challenges may face heightened susceptibility to various health conditions, underscoring the necessity for targeted interventions. These insights foster the potential for holistic health initiatives that prioritize not only genetic predispositions, but also environmental context. Public health campaigns can be tailored to emphasize the importance of maternal health, nutrition, and stress management, recognizing their long-term effects on offspring. Educational institutions might integrate epigenetic principles into curricula, promoting awareness of how lifestyle choices can shape health trajectories. Such knowledge encourages personal responsibility and informed decision-making. The ethical considerations surrounding epigenetic research also warrant attention. Issues of privacy, consent, and the potential for genetic discrimination emerge, necessitating careful regulation. This evolving understanding of genetics emphasizes the interconnectedness of individual choices and collective well-being, challenging societal norms and inspiring a re-evaluation of health paradigms. The implications of epigenetics extend far beyond individual health, influencing the genetic legacy passed to future generations. Alterations in gene expression resulting from environmental factors, lifestyle choices, and stresses create a ripple effect that transcends the present. This region of study reveals that experiences, both positive and negative, can modify an individual's DNA, potentially affecting offspring in profound ways. Inherited changes in gene regulation can lead to altered susceptibility to diseases, behavioral traits, and even cognitive abilities. These modifications highlight the importance of nurturing environments and healthy lifestyles, as they may not only shape one's own health, but also that of descendants. In considering the long-term effects of epigenetic changes, it becomes apparent that the choices made today carry significant weight. Societal shifts towards healthier living, improved mental well-being, and environmental conservation can foster a legacy that promotes resilience and vitality in subsequent generations. The intricate interplay between genetics and environment invites a broader discourse on responsibility and stewardship. Each generation stands at a crossroads, armed with the knowledge that their actions reverberate through time. This understanding of epigenetics underscores the importance of collective health initiatives and personal accountability.